So in this video, I want to walk you through how an action potential occurs. Now remember, an action potential is how a signal is sent along the length of the neuron, so specifically along the axon. Ultimately, when an action potential reaches the end of an axon, something will happen. Usually a neurotransmitter is released, and that will either excite a different cell or cause that other cell to not do something. So it'll either inhibit or excite. But in order to understand that whole process, we need to walk through how an action potential occurs. Now, as you can see, I've already drawn out an action potential curve. So right here, we're at our resting potential, okay, which is about -70 in most neurons. All right, that's where, if nothing's happening, that's kind of its happy place based on membrane potential that we talked about earlier. So we've got leak channels that are open, we've got the sodium potassium ATPase pump, and we've got proteins that are close to the membrane. And remember, those proteins that are close to the membrane of the cell are negatively charged. Okay, ultimately, this contributes to that resting potential of about -70. Now, in order for the magic to occur, for that action potential to occur, we have to get to we have to reach threshold. And so threshold is either that all or none thing. And so something will happen, some sort of graded potential or small, relatively small change in membrane potential will occur so that resting potential goes from -70, or the membrane potential goes from -70 until we reach -55. If we reach -55 in a neuron, threshold has been reached, magic happens through the action potential. And so going from -55 to +30, this is depolarization. So I'm just going to abbreviate it DEP, so that's depolarization. Once we hit -30, some stuff happens, channels open and close, and so we hit repolarization. And so repolarization, which I'll write here on the inside, goes from this this +30 until we get back down to -70. And then from -70, we drop all the way down to about -85 before returning back to -70 and being at our resting potential. And we refer to this as hyperpolarization. Now, as I discussed a moment ago, resting potential is determined by two sets of leak channels, the sodium-potassium pump, as well as proteins. These things are still there, ions are still moving through these leak channels, the pump is still working, regardless of whether or not we're in an action potential. But there are other channels that are involved that determine whether or not an action potential is occurring, what phase, that sort of thing. And really, we've got two sets of voltage-gated channels. We've got a sodium and we've got a potassium voltage-gated channel. Now, the potassium one's really easy. It's got a single door. When that door opens, potassium can move through it. When it closes, potassium can't move through it. Sodium, on the other hand, is complicated, or a little more complicated. It's a double door. So like any building that you walk into on campus, you actually have to go through two sets of doors to get into the building. If only one of those sets of doors is open, you won't be able to get in or you won't be able to get out. You have to have both of them open so that you can move from one side to the other. And that's how it is with this sodium, with this voltage-gated sodium channel. Now, as I've got written up here, we've got an inactivation gate and we've got an activation gate. So both of those need to be open. Now, these gates for both sodium and potassium open and close at different times, and that will ultimately determine what ion is moving during each stage of the action potential, or another way to look at it is what ion is responsible for each phase. So we're going to start off here by saying rest. Now, there's not really an ion that's responsible for it because we go back to those leak channels and the proteins and the things that we've talked about previously. Now, during resting potential, the sodium inactivation gate is open. So O just means open, but the activation gate is closed. And so what this means is that sodium is unable to move through that voltage-gated channel. But like I said a moment ago, sodium is still moving through the leak channels and it's being pulled back out by the pump. Similarly, the potassium voltage-gated channel is also closed. And so there aren't any ions moving through these voltage-gated channels at resting potential. But once we hit resting, or once we go from rest to threshold, once we hit threshold, depolarization starts. And when depolarization starts, or what starts depolarization, is the inactivation gate remains open, the activation gate opens as well. And so now both of those doors are open, and sodium can rush into the cell. Now, we don't need a lot of sodium to move in because we're having small changes, relatively small changes in membrane potential. It only takes a few ions to actually depolarize the cell. But that activation gate is open, the inactivation gate is open, so sodium is able to go in. All right. Now the potassium gate opens, but it opens slowly. Okay. So there is some potassium that's leaking out of the cell through this voltage gated channel, but it's a very small amount. There is much more much more sodium going in. And so as a result, the ion that's responsible for depolarization is sodium, and it's sodium influx. Okay. Now up here at the X that I've made, that I've drawn, that's where depolarization ends and repolarization begins. Okay, and so repolarization starts at plus 30 and remains in place until we get to negative 70. Okay, and so what happens when we get to plus 30 is the inactivation gate closes, or in other words, at least the way that I look at it, is that we are inactivating depolarization. The activation gate remains open, but since we've only got one of those two gates open, sodium is unable to move through the voltage gated channel. Just to reiterate, and I'll say this a lot, it's still going in through the leak channels, but we're not going in through the voltage gated sodium channels. Okay? Now, at this point, the potassium gate is all the way open, and so now we can have a lot of potassium leave the cell. And so repolarization is caused by potassium efflux. All right. Now, when we get back down to resting potential, everything resets, but there's a problem, okay, or something that kind of holds everything up. And so we've got hyperpolarization, okay, and I'm going to put part one or first half, and then I've got second half, okay. Now, like I said, everything resets. So what happens is the sodium inactivation gate opens, the sodium activation gate closes. Now, what happens with the potassium gate is that it closes, but it closes slowly, okay? And so as a result, potassium can continue to leak out. Notice here that where I've got sodium, we've only got one gate open, so sodium's not really able to do anything with membrane potential because only one gate is open. However, we continue to have potassium efflux because those voltage-gated potassium channels close slowly. And so that gets us down to this part down here. When we get to negative 85, that's where that potassium gate closes all the way. And so now we'll just say closed fully. Okay, the inactivation and the activation gates remain the same. So the inactivation is open, the activation is closed. But as you can see, we're at negative 85. How do we get from negative 85 back up to negative 70? This has to do with potassium, okay? But this is the one exception, or one of the two exceptions that we'll talk about this semester. Instead of potassium continuing to leave the cell, potassium actually comes back in the cell, okay? And this is due primarily to the fact that, one, we have a lot more potassium leak channels than we do sodium, and two, those negatively charged proteins that are close to the membrane pull those potassium ions back in. Because of the electrical charge. Remember, positives like negatives, negatives like positive. A negatively charged protein would really want to be with a positively charged ion such as potassium. Okay, and so just a quick recap. We start at resting potential. When we go from resting to threshold due to some sort of graded potential, what happens is that sodium activation gate opens. That allows a lot of sodium to rush into the cell. When we get to plus 30, that sodium inactivation gate closes. The potassium gate opens fully, and so now we have a lot of potassium leaving the cell. And then we go back, we become more negative, right? That we're repolarizing or returning to our original negative state. However, when we get to our original negative state and everything resets, meaning the sodium activation gate closes, the sodium inactivation gate opens, that stops or continues to prevent sodium from going into the cell through that voltage-gated channel. The potassium gate closes, but it closes slowly, so some potassium is able to leak out. Once that door is finally closed all the way, then what happens is that the negatively charged proteins attract positively charged potassium, and positively charged potassium comes back in through the leak channels. So this is a basic overview of how an action potential occurs and what's going on with the different ions and gates.